Thank you for being a citizen science volunteer and participating in the Land Snails and Millipedes Transect. This video will serve as education and as a reference for your participation. Land snails, slugs, and millipedes are an easily overlooked and understudied part of the ecology in the southern and central Appalachians. Of the more than 1,000 snail species known to occur in North America, only 25 have been identified on the parkway, but another 125 are likely to be found here, some of which may be rare or even endemic. Land snails, slugs, and millipedes are all extremely important for their role in decomposing leaf litter that returns important nutrients to the soil. To determine where you want to survey, have a look at the map on the Hands on the Land website. Once you've made your selection of one or more sites, register with Hands on the Land and then email Paul Super at paul_super@nps.gov with a list of your desired sites by Blue Ridge Parkway milepost number. Fieldwork can be performed any day from April 8th to June 30th, 2017. Please keep your personal safety in mind and avoid going into the field on days where there are heavy rains, high winds, thunder, lightning, or extreme heat forecasted. For collection, you will need a clipboard, data sheet, plastic or glass jars and vials with ethyl alcohol, a metric ruler, permanent marker, magnifying glass, shaker box, paper towels, and a carrot. If you do not have any of these items, some can be offered to you. Items for collection will be distributed at the training events. If you are unable to make a training event, items can be picked up by coordinating with Paul Super after you register for the project. At the end of the project, all specimens and equipment can be returned to your closest visitor center. The collection sites can be viewed by satellite image on the Hands on the Land website. The collection sites are broken down by milepost. Some locations are at the milepost and others are located in between. Make sure there is sufficient room for your vehicle to pull completely off the road and allow for you and others to get out of the vehicle safely. Make sure to display your dashboard sign and place valuables out of sight. Before you begin your field collection, assess your location for hazards. Bare sticks, logs, and rocks on slopes can be slippery, especially when wet, and may be hidden by dead leaves, so watch your step. Standing dead trees are hazardous. If you remove bark from them, keep your eye on the movement of the tree, or if someone is with you, have them serve as a lookout in case the snag begins to fall. There is always the possibility of venomous snakes or black widow spiders, especially at lower elevation sites, so wearing some sort of gloves and not putting hands or feet where you haven't visually checked first is always appropriate. Look for a stick to poke around the areas first. When you reach your collection site, begin to look for habitat. Snails like moist areas, so look for piles of leaf litter, depressions, under logs and on the undersides of logs, under the bark of standing or down dead trees, cracks among rocks, among moisture-loving plants such as ferns, and in shaded areas or ravines having ample ground moisture. Before you begin collection, fill in the date, time, location, and other basic information on your data sheet. Most of the surveying will be done by looking in habitat and spotting empty shells or live specimens. Empty shells can be placed into your collection jar. You can use an old food jar that has been cleaned out or an old medicine bottle. Keep larger specimens separate from the smaller ones so they don't get crushed. When you come across live specimens smaller than 15 millimeters, place the specimen in one of your sandwich bags. You might want to go ahead and prep a few of your bags before you go into the field. Take a sewing needle and poke just a few tiny holes in your bag. Then place a small piece of carrot and a small piece of wet paper towel in the bag. This will keep the snails alive for at least a week. Since there are some carnivorous snails, it is best to keep the live specimens separate. Be sure to mark your field data sheet with the information from your collection and mark all collection bags and vials with the site number and date if there is room. When you find snails that are larger than 15 millimeters, you have the option of taking photos instead of having to collect the specimen. You will need a shot of the sides, the bottom, and the top of the snail. Place your metric ruler beside the shell so that the length can be determined. Make note of size on your field sheet. Body color is important as well, so try and get a shot of the body. Please upload your photos to the Hands on the Land website under your site description. Be sure to make note on your data sheet how many specimens you photographed. Since millipedes are found in the same habitat, you will likely stumble across a few of them as well. Millipedes move quite slowly, and many will ball up when threatened. Centipedes are faster and will bite if threatened, so let the centipedes go. Millipedes have 2 sets of legs on most body segments, while centipedes only have a single pair of legs per segment. Count the body segments, and on the 7th body segment, confirm the pairs of legs. If you find only 1 set of legs, look for the gonopods. And if you find them, you have found a male. Upon identification, male millipedes are dropped into a vial with alcohol for preservation. If you are uncertain, go ahead and drop the millipede in the vial. If you find millipedes several inches long and as thick as your pinky, just take a picture of it and let it go. The next part of the survey will require the use of a shaker box. Grab some leaf litter and throw it into the shaker box until it is almost full. Place the lid on the box and shake it well for about 15 seconds. Empty the matter that passes through the screen into your gallon-sized Ziploc bag. Continue this process until your bag is about half full. After you've worked your way through the leaf litter, you will then gather topsoil. Scrape the top inch of topsoil off of a small area and place it into your other gallon Ziploc bag. Before you leave the site, make sure you have all the fields of your datasheet filled out. Once you are finished with collection and finished with sorting, you can return your equipment and specimens to the nearest visitor center. Items should all fit inside your shaker box. Keep live organisms on top of other things so they do not get crushed. Lastly, send an email to Paul Super to let him know where and when the samples were dropped off so he can get them to our taxonomists. Thank you again for being a citizen science volunteer and participating in the Land Snails and Millipedes Transect. We couldn't do this work without you.